Great. Good morning, everyone. My name is Ted Tenoff. I'm a Managing Director, Senior Biotechnology Analyst at Piper Jaffray. Our next presenting company is Celyad, a Belgium-based cell therapy company. Celyad is publicly traded on Euronext in Europe and IPO'd on NASDAQ last summer under the ticker CYAD. Celyad has two primary cell therapies, CCure, currently in a phase 3 CHARTS trials for heart failure, and also a novel CAR product, NCAR2, in a phase 1 study in AML and multiple myeloma. Here to present the Celyad story is Steve Buckanavage, who's the Vice President of Marketing. Steve, thanks so much for joining us today.
Thanks, Ted, and hello, everyone.
So I'm going to ask Steve a couple questions, and I think he's going to sort of work through with some slides. So Steve, if it's okay, I thought maybe we'd start with CCure since that is the most advanced product. Maybe you can describe to us the phase 3 CHART-1 trial and when we should expect data.
Okay, happy to. So CCure really represents one of our therapy areas where we're focusing on ischemic heart failure. We have completed a phase 3 trial in Europe, and in fact, if I can advance the slides here, this sort of gives you a roadmap and our milestones that we expect this year. So this is the first time ever that we've have a cell therapy that's advanced to completing a phase 3 trial. Now, Maury showed you this morning, he put up all of the active clinical trials, and there was a lot of phase 1, phase 2. And I know he meant to say this, but he meant to point out that Celyad is in phase 3, and that we've completed a phase 3 trial in Europe. In fact, you may have read on the wires that we have disclosed we've filed a letter of intent with the EMA, planning for success, to go ahead and follow through with registration. So CHART-1 trial is complete. We anticipate that readout in June, and we've said that before at multiple meetings. So this is a big month for us and a big month for cell therapy. And on the back of CHART-1 for Europe, we have FDA approval to move forward with a companion trial, CHART-2, which we're going to focus mainly in the U.S. There'll be some other international sites, but mainly in the U.S. To focus for FDA and approval. So that's a big milestone for us, and we're very excited about it.
Excellent. And just as you mentioned, literally just last week you mentioned, or you reported that you'll be filing in Europe on CHART-1. So tell us sort of what that does for the company and how you're able to file on a single phase 1 or phase 3 study.
Well, it's a good-sized trial. I mean, it's 240 patients, and it's on the back of some solid phase 2 data. So if you look at some of our foundation stones, there's 3 words up there in phase 2 that really should catch your attention. The first one is stronger, second one is smaller and longer. What we observed in phase 2 is that ejection fractions actually improved, so hearts got stronger. We saw some cardiac remodeling. These ischemic heart failure hearts actually got smaller, so that was pretty profound to us. And then on a more of a clinical patient-related endpoint, we saw patients statistically significantly walk longer on a standard 6-minute walk test. So with that foundation in phase 2, that translates in phase 3. We feel confident that we have a package that's robust enough for regulatory approval in Europe, and that's what we're busy today planning on those positive scenarios.
Excellent. Well, I'm looking forward to that data.
We are too.
Now, with respect to CHART-2, so we'll start that U.S. study after the CHART-1 readout. Is that the right way to think about that? And how long would that take, and when can we get data from CHART-2?
Right. So how you should think about it is that we are going to start enrolling patients as quickly as possible after CHART-1. We've actually already started to engage in preparatory activities, again, planning for success with CHART-1. We would anticipate that trial to finish. It should take 2 to 3 years. It's probably going to be in the similar sizing as CHART-1, with then roll out to regulatory approvals.
And just since you are the Vice President of Marketing, maybe I can give you a marketing question. How do you look at the opportunity both in the U.S. and in Europe in terms of the heart failure market that CCure could treat?
Right. Well, you heard this morning from the panel the word transformative, right? And on our opening slide, we had transformative therapies because our aim is to deliver these therapies that actually restore the body's ability to repair itself. So when you look at a positive data set for CCure in CHART, this literally is a transformative event. So we have the defined market, defined by our target population of class 3 heart failure patients, but this opens up just pathways of clinical development for label expansion and to be able to move forward. So I think we're going to be establishing a new category, a new way of treating, and a new capability within centers in Europe and in the US in terms of heart catheterization and how these procedures are actually done. So we think this is going to be an exciting time, not only for patients, but for the whole industry, because we literally, with a positive data set, will have first mover advantage and pave the way to transform how ischemic heart failure is treated.
Great. So I want to devote the rest of the time of our discussion to the CAR programs and your natural killer cell receptor work. This is based on some technology that was in-licensed from Dartmouth. So maybe you can describe what NCAR2 is and give us an update on sort of where the phase 1 study is today.
Okay, great. I'm happy to do it. Let me just put this slide up to introduce the topic. So you just heard Ted say your NCAR program, NCAR-T program. Again, this morning, I guess it was a good setup for, at least for Celyad, for us right now, because Oz and Shell and others talked about there's different ways. There's other ways to come at this than maybe CD19 or the approaches that we've all read about. That's exactly what attracted us to the technology that was developed in Charles Sentman's lab at Dartmouth University. We call it NCAR-T because it's natural killer receptor T cells. We're transducing a natural killer receptor, NKG2D, into a T cell. We're weaponizing that T cell with a naturally occurring receptor. And what this gives us is a different way to target cancers. And what's different about it is we're using the receptor as a targeting mechanism, not an antibody. The receptor has affinity, or there's 8 ligands that bind to this receptor, so that opens up pathways not only into liquid tumors, but into solid tumors. We've got some compelling preclinical data that was very, very interesting in terms of survival rates and survival rates upon rechallenge, but we also have preclinical data in solid tumors as well, ovarian and colorectal and a couple of others. So as our clinical program, and we are in the clinic, starts to mature, we'll have a lot of opportunities to move forward very quickly. Now, let me just touch on this slide really, really quickly. Again, from a what's different and what do we add to the whole CAR-T category, this is really our view of what are the 4 mechanisms or so that would really transform our thinking around transduced T cells. And you know, you've had a chance to read through this, but we think that if you can get one construct to have activity along these 4 areas, you really have something. And for us, it does all come together in a weaponized NCAR T cell. We call it NCAR2, so natural killer receptor-2, which is the NKG2D product. You can see some of the commentary here around what it does and how it acts along these 4 mechanisms. We are active in the clinic at the Dana-Farber Cancer Institute with a phase 1/2A dose finding and feasibility study. We've enrolled 2+ cohorts, some patients in AML as well as multiple myeloma. And to date, as we sit here today, no new safety or toxicity observations have been made. So we continue to progress at pace. We're very, very pleased with that.
Great. And I believe the company said that we should have data this summer. So again, a very busy June coming up for Celyad.
Yeah, June, we also anticipate sort of the first part of that dose finding study. And our intention is to disclose the completion of that first phase, and then we'll move into an expansion phase, having identified a dose. That's the plan, but this is early days for us, and so we're just glad that we're enrolling patients and that we're moving forward.
Perfect. Now, you discussed the ability of NKG2D to actually target more broadly. So is there opportunity to move beyond AML and multiple myeloma, beyond hematologic tumors, and look into other solid tumor cancers? Is that sort of the plan, to take NK or NCAR2 into solid tumors, or would it be a different construct that you would envision doing?
No, it wouldn't be a different construct. It's this construct. And again, this was part of the key features that really attracted us to this technology. With one construct, and because the receptor binds naturally to 8 ligands, those ligands are present on 80% of cancers, on solid and liquid tumors. So as soon as we identify the dose and we're comfortable, we can move very quickly into solid tumors with this one construct. We don't need to go back to go and start over again, develop a new antibody, et cetera, et cetera. We can go forward.
Very cool aspect of the technology.
Mm-hmm.
Now, you announced a partnership with Institute Curie today.
Yes.
To help with respect to development of these NCARs. So maybe you can tell us about that alliance or partnership.
Yeah, and I might as well just tee up the last slide. Partnerships and collaborations are important to us. I think they're important to a number of folks in the category. And this is part of our plan. You know, our world-class science has come from the Mayo Clinic, from Dartmouth College. We're active now in the clinic at Dana-Farber, and now we're really happy to announce this morning a collaboration for NCAR T cells with the Curie Institute and Dr. Ahmed Garina's research lab. That's going to be focusing mainly on, solely on NCAR-T and on solid tumors. And so some of the early work to get us from where we are today into solid tumors. As many folks here in the audience will know, CAR-T is let's just say, farther ahead, or the engine's really moving in the US. In Europe, it's just starting to build momentum. As a Belgium-based, European-based company, we're excited about bringing this to Europe, but we're also interested in additional collaborations.
One of the topics with respect to CAR-Ts and natural killer cell receptors in general that was raised on multiple panels this morning was the potential to move from autologous into allogeneic cell sourcing. Celyad's received 2 patent allowances for allogeneic CARs, or NCARs. But I think it's broadly CAR technology. So maybe you can describe these and explain how the field could ultimately move from autologous to allogeneic products.
Okay. So just to clarify, not just allowed, but patent was issued. So the Celyad allogeneic patent was recently issued. And what this provides for is the production of TCR deficient T cells that express a chimera. And this is no matter the process and how they're developed. It's a TCR deficient T cell that's expressing the chimera. And that covers a wide swath of ground. It not only enables a pathway, so a pathway into cancers, but it also puts forth a pathway for Celyad into areas such as infectious disease and autoimmune disorders. So we're really excited about that. We're starting to have some conversations with some partners about that. There's a lot of folks that are involved in this space in TCR deficient T cells, so there's a lot of excitement and conversations going on about that.
Great, excellent. And I do want to keep us on time, so I'll see if there's any quick questions from the audience. And if not, Steve, I want to thank you very much for being with us today. Also, just a quick plug, Celyad is going to be hosting an R&D day focused on their NCAR technology on Thursday here in New York. So if you want to learn more about the specifics of the technology, that should be a really great event. And then we look forward to the data coming out this summer.
Thanks, Ted. Thanks for the opportunity.